Okay, so this is basically a picture of a cell going through mitosis. So today we're actually going to be talking about the cellular level. And so we're going to talk about some of the structures of the cell. So you need to be familiar with them. You should know them. And you guys, how many of you guys have already taken bio?
I took Bio 10 last semester.
Okay, so this should all be fresh, right? So you should know all of the structures inside the cell and everything like that. So you'd be good. You should be good to go. Alex says, my chat. There we go. Okay, so so this is basically a fluorescent staining of cell going through mitosis. So if you actually look at it, the green part is your spindle, the mitotic spindle. Your chromosomes are here in blue, and then your cell membrane is all in red. Okay, so so cell membrane. So the cell membrane is basically made up of phospholipid structures. Okay, and so if you guys remember from last week, we kind of talked about the phospholipid. So the phospholipid has what we call like a dual characteristic. Part of it is hydrophobic, and part of it is hydrophilic. So hydrophilic means like water. Hydrophobic means scared of water. So the hydrophilic head likes to be in water or next to water. The reason why it does that is because it's polar. It's polar in charge. So it has a charge associated with it, or a partial charge associated with it. So that hydrophilic head is composed of a phosphate and a glycerol group. Both of those moieties are polar. Or in the case of the phosphate, it's actually an ion. Okay? So the hydrophobic tail is basically fatty acids. So it's basically here, this is an example of a saturated fatty acid, and this is an example of an unsaturated fatty acid. What is the difference between a saturated fatty acid and an unsaturated fatty acid? You guys remember? Okay, so that means no, you don't remember, or
no, I don't remember.
You don't remember? Alejandro, do you remember? So the unsaturated fatty acid basically will have a double bond. So you have a double bond between two carbons. And so that causes like a kink structure, or double or triple bond. Okay. So the cell membrane is, as I said, made up of phospholipids. And the way it's organized is that the heads are going to be facing towards the liquid. So the extracellular fluid outside of the cell is going to be facing the polar head, or the hydrophilic head. And then the tails are going to be connected together. They're going to form what we call a phospholipid bilayer, meaning two layers of phospholipids. So now your membrane structure has a job. The job of your cell membrane is to basically to allow certain things to go in and other things not to go in. So it kind of serves as like a form of protection, okay? The fact that it's able to let certain things in and not others, that term is called semi-permeable. Permeable means that things are able to go through. If it's semi-permeable, means it's selective about what it lets go through. A lot of times this is based upon whether it's polar or non-polar in the case of your lipid structure, but it also can be based upon size. Okay. So since things are selectively let in, some of the ways that it controls this is by having proteins that are also in the membrane that will allow things that are charged to go in, right? Because, we know that since this guy doesn't like water, it's not going to like anything that's polar, right? So then it's not going to let anything through it that has a charge associated with it or that is polar itself. So this here basically is showing you the cell membrane, and the key components here is that there are going to be proteins that are embedded in the membrane. So some of them are going to go from one part of the membrane or the outer part to the other side. So those guys are called integral membrane proteins. Some of them are going to be peripheral, meaning that they just sit on one side or the other. Okay. So here we actually, in the cell membrane, you actually have your proteins, and we have like these guys are protein channels. So these channels will actually let certain things through, like for instance, aquaporin. Aquaporin is a protein that is an integral protein that serves as a channel, and the name kind of tells what it does. Aqua meaning it lets water in. Porin means like a pour for water, specifically for water. So aquaporin is it's a controlled or facilitated diffusion type of event that allows water to go in and also go out of the cell. Okay. So here we also have cholesterol that's embedded in your membrane. We have these are called glycoproteins. A glycoprotein is a protein that has a carbohydrate attached to it. And we also have glycolipids. A glycolipid is a lipid that has carbohydrates attached to it. So basically, it has sugar hanging off of it. Okay. Questions, concerns, Cash? Okay. So this here is an example of simple diffusion across the plasma membrane or the cell membrane. Have any of you guys ever broke wind? Never? Anyone? There's only two of you, so yeah.
Yeah,
Broke wind, or let's say farted or tooted, or
flatulence, yeah.
Flatulence, thank you. Yeah. So you guys all have, right? So initially, when you break wind, it's all concentrated around you, right? So that deals with that concept of whoever smelt it dealt it. You remember that?
Yeah, I remember
as
that.
a kid, right? So, but over time, people across the room are able to smell it. So that's an example of diffusion. So you're going from a concentration that is really high, and it's able to sit there. And since there's a lower concentration on the other side of the room, gradually that gas is going to diffuse to the other side. Okay. So similarly, when you have a high concentration of a small non-polar molecule, those guys are able to diffuse across the membrane. Now, diffusion, simple diffusion, does not require energy. Okay, and so all the diffusion that's happening is that you're going down a concentration gradient from a high concentration and to a low concentration. This shows it nicely. So here we have all the small particles that are concentrated on one side, and then as time goes on, we get a few of those particles that are going across, and then eventually you should get to a point of what we call equilibrium, where they're going to be equal on both sides. Concentration is going to be equal on both sides. Okay. Good.
Yeah.
Okay. We also have facilitated diffusion. So facilitated diffusion often does require some type of energy to kind of control that process, but it kind of works the same way. In this concept, we have a channel protein, and this channel protein allows charged particles to actually go through, or selected particles to go through. Okay. Channel proteins are going to be less selective than, let's say, a carrier protein, because a carrier protein is going to actually allow certain specific molecules to cross that barrier, or they'll bind to something and they cross over with something else. Okay? Are you with me so far?
Yes.
Okay. Okay, so your cell membrane is it's a semi-permeable membrane. So kind of like if we put a membrane in here, and in this context, we have a high concentration of the particles over here, and we have a very low concentration over here, right? So this guy with the low concentration of particles is called hypotonic. This guy is called hypertonic. So in this context, if we have all of this in a solution, or in this case, water, then what's going to happen over time is that the water is going to diffuse into the other side of the membrane. So the diffusion of water is called osmosis. Okay. So, and it's going to continue to diffuse until concentration on this side is going to be equal to the concentration on that side. So a lot of the water will come over here until you have the equivalent concentration on both sides. So in that case, this guy would be called isotonic, meaning that on both sides of the membrane, they're the same concentration. Okay? You're good? Okay, so in the context of a cell, we have these same terms. And we'll see what happens. Okay, so we have a case where we have hypertonic. Hypertonic is, again, where the outside concentration is greater than the inside concentration. So as a result of that, the water from the cell is going to be leaving the cell and going out into the environment. Where in the case of hypotonic, hypotonic basically means that the cell doesn't have as much, in this case, it doesn't have as much water on the inside, or it has a lot, it's very concentrated on the inside compared to the outside. So then the water is going to be moving on the inside. The water is moving in this case, the water is moving out. So in the case of the hypertonic, the cell is going to shrivel up. In the case of the hypotonic, the cell is going to burst because water keeps coming in and it's just eventually going to blow up. Isotonic is where you have equal amount of water going in as coming out. So that means that it's balanced. The concentration is balanced on both sides. Does that make sense?
Yes.
Okay. Yes. So if we put cells in salt water, it makes it hypertonic because you're looking at the concentration of salt. So water is going to be going out. So the cell is going to shrivel up, right? So if it's in your bloodstream, if you have a lot of salt in your bloodstream, which happens when you have a high salt diet, so then that's essentially what happens, is that your cell starts to shrivel up a bit, and it takes the water out of there, out of the cell, and you become what? Bloated in a sense. Okay, are you guys good? Okay, so we also have what we call pumps. So these are ion pumps. So this is a sodium potassium ion. So these pumps kind of work both ways. It moves sodium in one direction and it removes potassium in the other direction. So, and in this context, they're moving opposite than the gradient. So you actually, this is going to require energy. So it's going to require ATP to make this happen. So in this case, we have high sodium on the outside and we have a little bit of sodium on the inside. So the cell is not supposed to have too much sodium. So in the sense, it keeps it low. So it'll actually put the sodium in the channel and then it'll close up the channel and then let the sodium out, okay? And so, but this process, again, requires energy. So the sodium goes ahead and binds in here, and then ATP will bind, and then you'll get the release of the sodium. So you're pumping it out into a high concentration gradient, which is total opposite than what diffusion does. Okay, where in the case of potassium, so potassium is low concentration on the outside, high concentration on the inside. So what it does, potassium will bind into the same channel. And then when ADP is released, then it'll actually let in the potassium. Okay, again, requiring both of these events require energy because they're going opposite of the gradients that they are. Okay. So, and all of these are actually serving as transport across the membrane, okay? Another way of transporting across the membrane is called endocytosis. So, in the case of endocytosis, there are 3 types of endocytosis. There's phagocytosis, there's pinocytosis, and there's receptor-mediated endocytosis. So in the phagocytosis basically means cell eating. So it basically means that, or say phago means eating. So in the case of phagocytosis, the cell membrane will actually loop around and then form a vacuole where it takes in the whole particle, the whole big thing, and then bring that on the inside of the cell. So it will endocytose it inside the cell. And then there, it'll actually break down the particles. Lysosome will combine with the vacuole, and then it'll put in these enzymes to actually break it down. And then those elements that are in that big particle will be recycled and reused. Pinocytosis is basically you'll form these little small vesicles. Pino means drinking. So it's like cell drinking. So it just takes a little sip. So it takes a little small vesicle of this, and then whatever's inside, it brings on the inside. And then receptor-mediated cytosis basically requires a ligand to bind to a receptor, and then that causes the cell to close up, and then you get what we call a coated vesicle. And so this guy requires a ligand to actually do this. So often, if you guys think about viruses, so our cell, in a way to try to protect us against viruses, it'll actually go through phagocytosis and take that viral particle and just kind of suck it up. And then it'll get destroyed. In the case of sometimes with viruses, sometimes the virus, the structures on the virus will act like a ligand. So it'll mask itself like a ligand, and the virus will actually set on the receptor and also get endocytosed that way. So a lot of times the viruses will use these mechanisms to kind of invade the cell. Are you with me?
Yeah.
Okay. Okay. So, oops, I don't know what happened. Okay, so another form of transport is exocytosis. So if endocytosis is bringing stuff on the inside, what is exocytosis? Exo. Are you there?
Yeah. It's to leave the cell.
Yes.
Exit it.
Yeah. Okay. So we're basically allowing stuff to get out of the cell. And the way this is done, a lot of stuff are packaged up in vesicles. These vesicles then fuse to the membrane and then are released into the extracellular matrix. Okay. So this is also called secretion. So here's an example of a pancreatic cell secreting enzymes. Okay. So here you can actually see the little vesicles, the secretion vesicles being formed, and they're actually being released from the cells. So all of those enzymes will be released into the extracellular liquid or fluid. Okay, questions? Cash? Credit? Okay. So what are the forms of transport across the membrane?
Endocytosis, exocytosis, receptor-mediated endocytosis,
Okay.
phagocytosis, penocytosis.
Okay. What about those really small particles that are non-polar?
Oh, like diffusion,
Diffusion. Okay.
osmosis.
And then what about the ones that are polar? How do they get by?
Active transport? Oh.
They're active transport, yeah. So which requires energy, but or they also use what we call facilitated diffusion, right?
Oh, yeah, yes.
Okay. Oh, since you guys all took biology, you guys all know all of the structures of the cell. Mitochondria? What is the mitochondria?
It's like the powerhouse of the cell.
Okay, it's the powerhouse. So it produces our ATP, our cellular ATP. Cellular energy. Okay, ribosomes. That's right. It's for protein synthesis. The rough endoplasmic reticulum. Well, the rough endoplasmic reticulum has a couple of jobs. One of the main things that it's responsible for is also protein synthesis, but it produces membrane-associated proteins or membrane-bound proteins. So the way it does that, the reason why it's considered to be rough is because it has ribosomes on the surface. And so the ribosomes will actually translate the protein into the membrane structure. It also does some modifications to these proteins. And then also it tags them for transport to the Golgi apparatus where they get further modified. Okay, we have the nucleus. What does the nucleus do?
The control center of the cell.
Yep. So it's the control center. It's also where our DNA, which is our control in a sense, where our DNA is housed. So the nucleus is kind of like a structure for protecting the DNA, because the DNA tells everything else what to do, essentially. Or our genes tell everything else what to do by the production of RNAs and then proteins. So the nucleolus. So the nucleolus is responsible for ribosomal RNA production. So it's where your rRNA is produced. Ribosomal RNA, so the ribosome is not made up of proteins, it's actually made up of RNA. And so these RNA structures are what are responsible for building our proteins, your protein synthesis. So all of your RNA that are needed for your ribosome are made up in the nucleolus. So I'm skipping around because it all kind of has a way. So the Golgi apparatus. Okay, so the Golgi apparatus is for distribution. So distribution of proteins. It also is where proteins are modified. And tagging proteins for their destination. Okay. So the Golgi vesicle is just a vesicle that is used to, it's mainly used for secretion. Well, it's used to transport those proteins to their final destination, such as the vacuole or the lysosome or the plasma membrane. So there's also vesicles that come off of the Golgi and go back to the ribosome. I mean, not to the ribosome, to the endoplasmic reticulum. And so you also have communication between those two as well. Okay. So the vacuole is was basically shown in the other. So it's primarily used for it does a little bit of storage, but it also does it's also a spot where things are broken down at. So particularly those viral particles and stuff. So what'll happen is the lysosome will fuse with the vacuole and break down if it's a viral particle. Okay. So storage. and break down. Peroxisome, so they're basically to get rid of all your toxins. So particularly hydrogen peroxide and oxide toxins. So secretory vesicles, they're just vesicles that are used for secretion. So the smooth ER. So the smooth ER is smooth because there's no ribosomes on it. And it's also for de novo biosynthesis. De novo means of new. So things that are going to be made brand new. So like from the vacuole and from the lysosome, old proteins are broken down, old lipid structures are broken down. And because of that, it's recycled. So then we're able to use those amino acids all over again for other things. Well, these guys will build things from scratch. So they'll build an amino acid or they'll build lipids. So primarily your phospholipids are being made here, but it's capable of doing other de novo synthesis. But mainly the phospholipids that are used in the liver. The lysosome is responsible for breaking down and recycling those bio monomers that we use, those bio macromonomers that we use. Spell monomers. Okay, these guys make up your cytoskeleton. So your cytoskeleton is kind of like the cell skeleton. So they're for structure and then also for movement. The centrosome is it is the spot where your centromere will actually start to form, and then you get your spindle formation occurring from that area. So it's a kind of permanent structure that is used for when your cell is getting ready to divide and to copy its DNA or separate the copies of DNA that are being made. Let's see. And then, of course, we know that the plasma membrane is for cell protection, and then also for allowing the transport of selective things in and out of the cell. The cytoplasm is like the fluid inside the cell that all of your organelles are kind of sitting in, and they're also responsible for providing some nutrients and stuff like that, and means of transport of things. Okay, questions, concerns, Cash? Credit? Oh, good. Okay, so the endoplasmic reticulum, we've already kind of talked about it. We know that its job. So we have two forms. We have the rough ER and we have the smooth ER. So the rough ER is kind of surrounding the nucleus, and they form these little membrane sacs, which you guys can see up here. And so, and here's the smooth ER. They kind of have this little mounds, and so they're quite different from each other. So we know that the rough ER is responsible for protein synthesis, particularly of membrane-associated proteins or membrane-embedded proteins. The smooth ER, like I said, is responsible for phospholipid synthesis, steroid hormones. It also is responsible for the regulation of the concentrations of calcium. It's also involved in sugar metabolism. And it can break down certain toxins, not all toxins. The Golgi apparatus. So we know that the Golgi apparatus is like our distribution center, kind of like the Amazon hub, right? So all of the companies send stuff to Amazon, and then Amazon ships it all to all of us, right? So it's kind of like that. And so it's going to get stuff from the rough ER by a vesicle. So a vesicle is going to come deliver the package. Then in those vesicles, then it's going to actually start. It's going to start separating it, depending on where it's going to go. So some of it's going to go to the plasma membrane, some of it's going to go to the lysosome, some of it's going to go. And so the Golgi apparatus is basically like I said, the distribution center. It tells everything where it's supposed to go. Also, the Golgi, at a point when it develops, will actually produce the synthesis of the lysosome itself. So if the lysosome needs to be replaced, it'll actually replace it by generating all the proteins and the membrane that's associated. Okay, so let's see, what else should I tell you? So the lysosome is responsible for, as I said, breaking everything down. So it's just basically, it's a whole bunch of enzyme proteins that are responsible for destroying and clipping. Okay. And so hence lyse means break. So lysosomes, breaking, it's an organelle that breaks down stuff. Okay, mitochondria. So the mitochondrion is basically, it's a double membrane structure that's responsible for the production of our ATP. Okay, so cells in theory are capable of producing energy without the mitochondria. But in the case of, let's say we have a single sugar molecule, right? In a single sugar molecule, our cell is only able to produce 2 ATP without the mitochondria. With the mitochondria, is able to produce 32 ATP. So in terms of if you want a lot of energy, and we need a lot of energy to keep everything going, we need something like the mitochondria. So the mitochondria was thought to be a bacterial cell that was engulfed by our cells, or by eukaryotic cells at one point throughout history. And since it was engulfed, right, over time, what happened was that the mitochondria has its own DNA. And so it kind of can exist almost independently. However, over time, what happens is that the DNA, the mitochondrial DNA, certain genes of the mitochondria has moved over to our genome. And so to kind of keep it hostage, keeping it from jumping out or leaving, we've taken some of those genes and put it into our genome. So it requires us to kind of live. So we kind of live symbiotically. So it can't live independently because it needs some of those genes. Okay, so it's a pretty nice little theory, and then they've done some sequencing that suggests that that's what really happened. So there was an evolution that happened over time. So at one point, that bacteria was independent of us. So pretty cool. Peroxisomes. So peroxisomes, again, membrane-bound organelle, and they're responsible for basically detoxifying the cell. So it gets rid of all those toxins and stuff. Again, primarily peroxides and harmful oxides. And then they also do a little bit of lipid metabolism. So our cytoskeleton is kind of composed of three components. We have microtubules, we have microfilaments, and we have intermediate filaments. So microtubules are basically made up of tubulin. So they're like coils of tubulin, and they're like diamonds of each other. We also have actin filaments that are the microfilaments. And we have keratins. So, and these guys are just like fibrous proteins that kind of twirl around each other. So these guys are all responsible for keeping the cell shape. They help with cell movement. They also help with endocytosis. So that little loop for phagocytosis involves the projection of these guys, or the polymerization of our cytoskeletal units. And then they also aid in cell division. So when we're actually separating the chromosomes and actually going through the division itself. So the nucleus, like you said, was the control center of the cell. It contains all of our genetic material, or all of our DNA genetic material. RNA it starts off in the nucleus or in the nucleolus, and then is shifted out through the nuclear pores. So there are certain cells that kind of develop specialized function. So like, for instance, we have muscle cells. We have multinucleated muscle cells. Now, do you guys know what multinucleated mean?
It'd be many nucleuses.
Many nucleuses. So what happens over time in these cells, so we have our first cell, and this guy has a nucleus. So when it gets ready to divide, normally what would happen is that this guy would copy its, skipping steps here. I'm not going to give you all the steps. During mitosis, it would actually copy its DNA, make 2 copies and separate. And so you'd end up with like 2 DNA in the cell. And then it'll go through and split itself into 2 cells. So this is like your mother cell, and then you get your daughter cells. Okay, these guys, instead, they don't separate, they continue. They do this. They just continue. Then they duplicate again, and they get a little bit bigger. And then they duplicate it again, and they get a little bit bigger. And so over time, what happens is that they start forming these fibrous structures. And so you'll get these long fibers, these muscle fibers. Okay. And so these guys are long fibers that are going to be associated with, and they have tons of nuclei associated. Okay. And this basically happens after a very long time of going through the process of division. Okay. So you get, in a sense, these guys are fusing together. So instead of separating in a natural separation, they stay together, and they continue to stay together. They continue to stay together. Okay. Okay. See. So in the case of red blood cells, so red blood cells basically gets rid of its, so as it matures, at the beginning, it actually has a nucleus. So, and then as it gets older, it'll actually get rid of the nucleus, because the nucleus takes up a lot of room, and it wants to use all that room for hemoglobin. Okay, so it makes all the proteins it needs to kind of survive, and then it just kind of extrudes its DNA entirely. So it just ejects it. Okay. So only, yeah, so only muscle cells typically do this. So those muscle fibers, are doing this. So you guys, have you guys cooked chicken before?
Yeah.
We've all eaten chicken, right? So when you eat chicken, you see those nice little strips of chicken? Like you can actually break it off into little strips and long, thin strips.
Yeah, almost kind of like string cheese almost.
Yeah, yeah. So it's, yeah, thank you for a great analogy. I have to use that. Thank you. So in that context, those are just the muscle cells and those fibers. So that's literally one cell with a whole bunch of nuclei that you're eating. Okay, so the DNA, we've talked about this last time. So this is basically a strand of DNA, so you get strands. So the DNA actually forms a double helix, so these strands come together to form our double helix. Well, these double helix are then wrapped around what we call histones. So these histones regulate the genes. And so they allow genes to be turned on and to be turned off by keeping them wrapped around them, or when they need to be turned on, they'll kind of open up, and so they'll unwind around that histone a little bit and open up. So histones are basically used for coiling those DNA. And if you get a whole bunch of histones together, they form what we call the nucleosome. And so they're just DNA coiled around the histones. Okay. So then the nucleosome will form the chromatin. So chromatin is the way DNA is normally stored inside the nucleus. So it's stored as chromatin, so it's all connected together, not separated. So as soon as we get ready to go through mitosis or meiosis, so we start forming chromosomes. So the only stage that we use chromosomes is when we're going through the mitosis, when we're getting ready to go through mitosis or meiosis. Okay. So, and then that's where your DNA will actually separate into each of the different chromosomes. Okay, so you guys are already familiar with the DNA structure. You guys remember that we have a nitrous base, we have a sugar and phosphate backbone. And that's what you guys have to see here. And the adenine bonds with here, or forms a hydrogen bond with a thymine. So A matches up with T. And guanine forms a hydrogen bond with cytosine. And so G matches up with C. Okay. Questions?
No.
Okay, so DNA replication. During DNA replication, there are proteins that are involved in that process as well. So, we have what we call a DNA polymerase. So do you guys, poly means what?
Many.
Many. Polymer, mer means unit. So many unit. So a DNA polymerase will actually combine each of the nucleotides together so that you're making, from each individual nucleotides, we're making a polymer. We're making a polymer of DNA. So DNA polymerase is the enzyme that's responsible for covalently bonding each of the nucleotides together. Okay, and so that's essentially what goes on. So it uses one strand. One strand is considered to be the template. So that's the original DNA strand. And so the other strand is the one that's being synthesized. So the DNA polymerase will actually grab the nucleotides and attach them covalently to each other. as it goes by. Okay, the DNA helicase, the helix, so it actually will split the helix. It will unwind that helix so that the polymerase can actually work its magic and duplicate the other strand, or make a complement to the other strand. Okay, we also have what we call the lagging strand. The lagging strand is actually going in the opposite direction. So this guy unzips the helix. This guy will actually go in that direction that is being unzipped. Lagging strand, it starts off, you have what we call an RNA primer, right? Okazaki fragment. And then it'll actually start from there and start producing DNA until it reaches to the butt of another one that it just started, and so on and so forth. Okay. Okay, so the process of going from DNA to RNA, so the DNA is actually going to be serving as a template. An RNA polymerase will come in and bind to that DNA template and then make the RNA. So then the RNA, that process going from DNA to RNA is called transcription. So when you're making the duplicate of DNA, called replication. So from DNA to RNA, transcription. And so then your messenger RNA is then exported to the outside of the nucleus to the ribosome, and then it's translated into a protein. Okay, so your RNA, your DNA is kind of like being copied, so it's being transcribed, right? So transcription. Then your RNA, that transcription, right? That RNA is then being translated. So you're going from RNA language to basically, or nucleic acid language to protein language. So it's being translated. Okay. Are you guys good? And so then we get our proteins, and that protein does its specific job, right? Okay, so this is where we stopped in the other class, isn't it? Let's see.
Yes.
Number 26. Yeah. Okay, so we'll talk about the details of DNA to RNA in more detail. Like I said, well, it's already here. So we have the RNA polymerase that does the same thing as the DNA polymerase, the only thing it makes RNA. So it uses that DNA as a template, and then it actually will transcribe that RNA fragment. So then we'll end up with an RNA transcript. This RNA transcript is called messenger RNA, or mRNA. And we're going to stop right here. It's so easy with just two people, and it goes by so fast.
Yeah.
Okay, are you guys good? Let's see, what time is it? Man,
Yeah,
not even an hour. Wow. Okay, so we'll start with, are there any questions?
Not at this point.
Okay. So yes, you have a question, Alejandro? Yes, that'll be fine. Yeah, you can cut it into a note, your notebook into paper. That's fine.
Oh, I've got a question on that. I have my printer that prints out cardstock, so I would usually make the flashcards on the computer anyways. Because I already submitted the assignment, because the way that I submitted it is not how I would like print them out, because they'll, like, on one side it wouldn't like match up, so I made them all match up, like left is the word and right is the definition.
Okay. Yeah, that's fine.
And then it was 10 flashcards for each chapter, right? Or is it 10 total?
Yep. Yeah, 10
Okay.
flashcards for each.
Okay, cool. I put in 20.
I guess I need to specify that. Remind me. Okay, so pretty simple so far. It's all kind of review.
Yeah, it's coming back slowly but surely.
Yeah, it's good. Okay, so then I will let you guys go, and I will see you guys next week.
Okay. See you on Monday.
Okay,